Welcome to ASM Live, the internet talk show live from the 2012 General Meeting of the American Society for Microbiology in San Francisco, California. I'm Stanley Malloy. I'm the host of ASM Live. I'm the chair of the ASM Communications Committee. I want to, first of all, encourage you please to ask questions throughout. If you're watching online, you can use the chat function on the video viewer, or you can send questions over Twitter through hashtag ASM2012. Or if you're here in person, we also would love to have questions in person. In this session, we're going to be talking to Forrest Rohwer and Lita Proctor. And we're going to be talking about a session that they're running at the meeting called When Good Bugs Go Bad, Microbiome Dynamics and Disease. Lita, could you tell us a little bit about this, about the session?
Sure, I'm happy to. And it's Lita.
I'm sorry.
That's okay. Sure. Forrest and I last year started brainstorming about a microbiome session. Of course, the Human Microbiome Project is a really large, visible program in the human microbiome arena, but we really wanted to feature things that the HMP wasn't doing. And so we started brainstorming about that and thought about maybe trying to put the microbiome more of an evolutionary context. So to bring in people who work with non-human systems and to understand sort of the evolutionary context of what the microbiome is.
That's great. Yeah. So Forrest, could you tell us something about, you're going to give a talk in this session. What's the theme that you're going to talk about?
Mostly the competition between different coral holobionts and algal holobionts. Mm-hmm. That's the goal in this one.
What is a holobiont?
A very controversial term, but so we use holobiont to mean the organism, like the coral animal, plus all the bacteria and the viruses and so forth that form an ecological unit on a coral reef, and then they holobionts actually compete with each other for space on a reef. But it applies to broadly to any macroorganismal and microorganismal assemblage.
So what are the key things that you've learned in these studies?
Yeah, so most of what we initially had focused really on the how there was more classical symbioses in a mutualistic sort of sense of the word. So how, for example, a coral will recycle nitrogen by using the coral animal, archaea, bacteria, fungi are all involved in the recycling of nitrogen on corals, which is important because corals live in places where there's not enough organic nitrogen. Nowadays, it's much more about a lot of what happens when the symbioses start to change in such a way, usually associated with human endeavors, leads to where an algal holobiont can walk over the top of a coral holobiont and actually kill it off effectively. And there's definitely a microbial interaction component to that.
So the human endeavors stimulate this process, which in fact results in the demise of the corals.
Yes, yeah, that's what it is for the most part.
Yeah. And viruses play a key role in this process as well?
Yeah, there's many things that happen with the viruses that are fun. There are, of course, just the viruses that they seem to get stimulated by the stress. So you have a coral sitting next to an algae, and just the stress of that interaction will induce herpes-like viruses out of the corals. So that's one of the things we see. But there's, of course, all the phage dynamics. So the phage are bringing in different genes that are important for the working of the system.
So often when we talk about microbiomes, different places, we think of some place that's reasonably easy to sample, easier than other places. But when you think about how you would sample those communities in the marine environment, where you have things flowing in and out, it seems pretty complicated. How do you do that type of study?
Well, on a ship, on scuba gear. No,
Well, you swim around a lot with big carboys.
Exactly. Yeah. Lita always laughs because she says I get paid a lot to collect water underwater, which is kind kind of moronic, but yes. No, so so the logic of actually working on coral was to avoid everything that you're talking about. It turned out to be horribly complicated, but the logic at the time was it was really hard to go back to the same piece of water. And so we thought that a mucosal surface would be making itself, so then you'd have a standing microbial community and that it would be easy. It turned out that it was much more complicated than we thought, but it's been pretty fun. It's a great system to work on, even though it's complicated.
And are there lessons from this system that are directly applicable to some of the other types of systems?
Yeah, this is, people don't remember, but most of the microbiome stuff was originally done on corals and related organisms. It basically started with marine biologists and then moved into the human field.
That's right.
Yeah. In fact, you couldn't get money for the human biology when you could get it back in the early 2000s.
It's amazing how that changes.
Yeah, exactly.
But hopefully, though, there is going to be some kind of a cascade effect, where this stimulates new areas of research. The NIH Human Microbiome area will stimulate research in the other agencies like NSF to support microbiome-related science. I think that's already starting to happen.
Yeah, definitely.
I think that's an important lesson for people, is that sometimes the best way to get an understanding of humans is by understanding non-humans.
Absolutely.
And it's really been one of the fundamental concepts in basic research that's driven the U.S. forward. So Lita, there are a number of other talks in this session that deal with different types of host microbe systems. Could you tell us about a couple of them?
So one of them that comes to mind right away is use of Hydra as a model system for I can call them alternative model systems for probably application to human microbiome. And we have a scientist, Tom Bosch, who's coming to give one of our longer 30-minute talks on that, and utilizing Hydra. I guess they developed some kind of tumors, and so using those Hydra as a model for cancer in human systems. That's one example. Another example is there's a talk that will be given. In fact, Karen, I was hoping would join us here today, but Karen Guillemin, who's been doing a lot of work developing the zebrafish as an alternate model system for studying the human microbiome. It's a very nice system, transparent species. They reproduce very fast. They seem to be a very fairly easy model system to work with. So she's done a terrific job developing that as a model system for the human microbiome. A third one that comes to mind right away that I don't know as much about is there's a lot of interest, as Forrest has, in non-microbial, non-bacterial members of any microbiome. So we have someone giving a talk on the DNA virome. Virome being, all the viruses associated with the larger microbiome. So quite a diversity in the talks we'll have in our session.
So the title of the session is When Good Bugs Go Bad. Now, you've given us a little perspective relative to corals. What about in these other systems? How does that tie in?
Well, of course, as usual, for any ASM session, we want it to be catchy and sort of grab people's attention. So we were sort of brainstorming about what's the coolest thing we could talk about. But also, I just mentioned Tom Bosch. He's going to be talking about Hydra and these tumors they develop, and that's a model system for cancer. Of course, that's a good bug system gone bad in some form. So I think in most cases, I think all of our talks have something to do with some relationship to human disease. Of course, when good bugs go bad, it doesn't necessarily lead to disease, but I think that's the main kind of central focus around the talks.
Yeah, it kind of matches that idea that you're seeing where, the microbiome shifts basically not always membership, but just the relative abundances of things. And that seems to be in that gradation of symbiosis from mutualism to pathogen. They're very connected, and you can find that in humans and all across the natural world, where something that normally lives with the organism is what kills it in the end. So there's a reasonably good chance we'll die of microbes we're carrying right now, right? And they'll eat us when we die. So it's.
So I want to ask Forrest a question.
No,
because I mean, I'm sort of peripherally aware of what's going on in the symbiosis field. There's a lot of revolution in the field and sort of, expansion of terminology and so on. But you've decided to adopt the term holobiont, right?
Yes.
So help me understand better about when you approach this understanding of it, why you adopt that term and why that's the way to describe the system you're studying, and how that applies. I mean, then of course how that applies to other microbiome systems.
Right. So it's actually mostly by precedence. So that the great thought thinker in this was Lynn Margulis back in the day, and she was thinking about first the endosymbiotic theory, and then trying to put together symbiosis in general, and she was trying to come up with a term that wouldn't apply positive or negative, but would just imply the collection of organisms. And instead of calling it an assemblage, she called it a holobiont. And so it's a symbiont, but it's all of the symbionts together.
Well, you originally said it was a controversial term, so I want to hear more about why you think it's controversial.
So it's controversial mostly because because of Lynn and her connection to Lovelock to all those people. So there's a new age-ish version of it, and then there is a not new. There's a scientific version of it.
I see.
Okay, so it as an ecological assemblage is pretty hard science, but as a evolutionary assemblage is controversial because people have a hard time understanding how you would get evolution with an assemblage as opposed to a population of organisms.
Oh, I see. Okay.
Yeah, so you can imagine, and if you get too wishy-washy with that, the evolutionists will go for you, definitely. Yeah.
I see. Okay, great. Thanks for explaining that.
So I'd like to encourage questions from our audience and from our viewers out there in the internet and on Twitter. Please send your questions to us and join in on the conversation. So one of the things that you mentioned, Lita, see, I've got that right now.
Thanks, Dan.
One of the things that you mentioned was the virome, all of the viruses that are involved in these communities. And I think that a lot of people think that viruses never do anything beneficial, that if there's a virus around, it's bad, right? But the way you've described viruses in these communities, it's not all bad.
That's right.
It would be interesting to hear some perspective on that.
Well, Dr. Rohwer here is Mr. Virus, so let's hear from him.
I mean,
how would you say that?
It's all a matter of perspective, right? So there are definitely great examples of where viruses bringing things in that bring in genes, into systems that change the behavior very dramatically of the microbe. And that can be beneficial, and of course it can also, they bring in a lot of pathogenicity factors that we think of. So it depends on how you're looking at it, I guess. But yes, there's a number of really interesting examples. My favorite is the one, there's a fungi that lives with these plants that live next to hot springs, and it turns out that the gene that allows them to do that is that it's a virus that infects the fungi that then infects
gives them
the
some
plant.
kind of heat
it gives
tolerance?
them
interesting.
So you can imagine, actually, let's just call it niche expansion because of the viruses coming in and out.
I mean, there was a recent paper that just came out this past year about there's clearly some kind of a protein that the origin is from a virus that plays a huge role in placental development in mammals, right?
Yeah.
I mean, that's an example where apparently over millions and millions of years we're constantly being swamped by viruses, and periodically there's some property they bring to then allow adaptive advantage for the host.
Yeah.
So that's a great example from this past year.
But I think these really are examples that have come about because of studies of the microbiome writ large.
That's
where
true.
we've been able to see things that we couldn't see 10 years ago.
That's absolutely true. I also think that, I know this is a very educated audience, but in general, I think even just conceptually, the idea that the vast majority of microbes we come in contact with on a day-to-day basis and throughout human history have been beneficial microbes, even though we often think of, bacterial scourges and so on. In fact, the vast majority of microbes we come in contact with have been beneficial and are, key to supporting our own health and well-being. I think that concept, it's starting to really permeate even the kind of science that we practice.
I think you're absolutely right, except I would add a caveat.
Okay.
And that is that we have said for so long that these microbes are beneficial, but often we didn't know exactly how.
Right.
So you can easily get on Google and say, beneficial E. coli.
Sure.
E. coli is one of the organisms in our gut. How is it beneficial? It aids digestion.
Yeah, you know,
Well,
it's very
what
vague.
does that mean,
Right.
And is it true? And how do we know? And I think that the microbiome studies are providing ways of doing direct tests of some of these ideas.
It's also forcing us to consider not the single microbe, but the full community. So you talk about beneficial microbes, you can't just talk about a single individual, because there isn't one microbe living in your system. It's a whole community, a whole group that they're interacting, and that's the beneficial properties they bring, whether it's to the human or to any organism on the planet, has its own associated microbiome. But it's not going to be a single strain, it's going to be this community. I think that's the other sea change in the kind of science you see being practiced. We're looking at these microbes as communities.
So one of the big changes, the sea change, if you will, especially for corals, one of the big changes is our ability to accumulate all these DNA sequences and see who's there. But obviously that's not the only thing. In your discussion of corals, I think you brought up some approaches that allow you to complement those studies, to do ecology in a natural environment in a way we couldn't do a short while ago.
Yeah, I mean, that's the hard thing for people to get, that it's not just about sequencing. You actually have to put that in a really larger context, and that's, of course, very hard. But you do actually have to do the ecology and the evolution of the system that you're working in. And we see it all the times in human disease. The system, my favorite system, is cystic fibrosis, and that's a system where it's clearly you've got the evolution of the microbes within the system, but you've also got this ecology of the human host talking to the microbes and vice versa. And you can see that nowadays. We have the tools to actually ask, what is the human doing? What is the microbial community doing? What's the viral community doing?
So I don't know, I'll ask you, Forrest. So I'm trained as a microbial ecologist, formally, but I've been at NIH for a while, and I've been actually quite pleased to see more and more interest in embracing microbial ecological principles in studying disease. Now that's coming from the program side. Are you seeing that as well?
Not
as much as you'd like.
Yeah, the scientists were harder to get to go that direction. It turns out that the doctors are not. The doctors in general are. They already have to think that way. So they're a little bit holistic in nature, and so they're trying to figure out anything that will allow them to treat their patients correctly.
So they're more open.
So they're a little bit less reductionist and a little bit more wishy-washy, which sometimes in microbial ecology you have to take, because you don't know exactly why something is happening.
Sure.
So that's been my experience. And we are seeing a lot of changes in the way the scientists are looking at it too.
So how would you try to help bridge that more? In other words, you mentioned that the doctors are already open to it. Now you're talking about the research scientists, right, are not as open. And you've got the microbial ecological community. How do you think we should try to bring them together to try to talk to each other?
I have nothing useful to say about that. No, I think you must.
Sure you do. Of course you do.
I mean, the way
that's why I asked the question.
The way we do it is we just work with the doctor, but it takes a lot of effort by both parties. So we have a doctor. He's always at our lab meetings. He goes. He spends all the time there.
Wow.
Yeah, and
this is on your cystic fibrosis
exactly.
project. Okay, yeah.
And so and a lot of the people, remember the human microbiome, like Jeff Gordon and David Relman, are actually doctors, right? They started as MDs,
as scientists, right?
Yeah. So they've been really leaders in taking this forward.
But Forrest, it seems to me that there's another change that's been associated with this. So once upon a time, microbial ecology was, to a very large extent, a look and see type of field. Evaluate what's out there, and then try to tell stories. That it was very hard to do direct tests, to be a reductionist and a microbial ecologist at the same time. But these new techniques allow you to go in and sometimes ask reductionist questions in a holistic environmental context. And so I think that that is going to bring along a lot of us reductionist scientists who like to think in that way because that's the way we've been trained. And so in addition to the physicians who may already have this perspective from their training, I think that scientists will come along very soon. One of the key things that happens with scientists that you discover very quickly is where there's money, there's scientists. And when it moves, the scientists move.
Sure.
But because it takes money to do good science, and people are there because they love doing science. Right?
Yeah, that's completely true. And in fact, Lita was one of the leaders in the thing that actually, I think, transformed the field. And that was the Marine Microbial Initiative back in, when did that start? Like
In the Moore Foundation days, right? Yeah, right.
Because that was really, that led directly to what we call shotgun metagenomics. That's what paid for it. It was the first time that we could use next generation sequencing because before those sequencers were even available, we had the money to go and get people to let us use them. And so that was a big deal. And you had people that were doing ecology as well as all the new molecular tools.
So give us the 30-second version. You said shotgun metagenomics, next generation sequencing.
Right.
Put that into perspective. What is the change? Why was that so important?
Because we had no way to go and look and actually, so imagine studying, doing a study of a forest, right, without being able to see the trees. And that was kind of where we were at.
That's right.
And those things totally changed. We could go out and now we're actually way ahead of the other parts of ecology in many ways because you can do a community analysis and still see what's going on at the population, and you can know what's going on functionally because you can look at the genes that they're doing, and that's all incorporated in one data set.
That's right.
And then you can put all the other, what they call metadata surrounding that and do pretty amazing stuff.
So when you say we are ahead of the other ecologists, you mean microbial ecologists.
That's right.
Yeah, there's a lot of, it's very different, right? So someone that's been counting all the trees and measuring them in Harvard Forest for the last, let's say 100 years or something, has never looked at, 10 billion organisms at the same time, right, in the same way that we do.
That's an interesting comment because one of the key things that your session, I think, emphasizes is that, in fact, the communities are communities of small things and big things together. And the functionality of the community is dependent upon all them all together. Yet, you segregate out your area of microbial ecology from ecology writ large.
Yeah.
But whereas in reality, it's probably microbial ecology is a fundamental part of all ecology. How about that?
I would buy it.
Yeah.
I would buy it, except it's been a hard sell. So I think maybe that's another area that needs some kind of, collaboration and discussion and to see whether the macroecologists really think that the microbes in their systems they studied are really the key members of that community. I think that's really another conversation that has to happen.
Yeah, and there's also a mistake, I think, that the microbial ecologists are making, which is that we're just applying macroecological theory to microbes, and it's not. We should be developing the new theory.
And there are new theories that are being developed. Once they realized you couldn't really utilize macroecological principles, then new ones are being developed.
Right.
Yeah.
And I think those will take over actually regular the macroecology, because I think you can look at a different level that they've never got to.
So one of the issues with respect to developing those types of theories is it demands people with a very deep perspective in mathematics as well as biology. And that's another, I think, area of emphasis that's changing in science. Right?
Well, there's a huge growth because of sequencing technologies, maybe not the only reason, but one of the main reasons. We're seeing a huge influx of students who are very competent in computational skills, in mathematical skills, and statistical skills. And so I think we're seeing this real enrichment of quantitative scientists entering the field of biology, which often hasn't been a quantitative science. Now I think it's becoming very quantitative. So I think we're going to see a real change in that.
Well, the last big time a lot of quantitative scientists moved into biology was the birth of molecular biology.
That's right,
which
that's
clearly
right,
has
that's
had a
right,
big
exactly
impact.
right, exactly right.
So I think that this has the opportunity to be the type of revolution that will be stimulated by another surge of quantitative
I think
biology.
so, yeah.
So this is, well, so this has been a great conversation. Lita, you are also going to be involved in a session a little later. Could you tell us about what the next ASM Live that you're going to be involved in is
Well, I don't know if it's an ASM Live, is it? I don't remember that part, but we're having, so ASM has this terrific idea of late-breaking sessions to really capture something that's phenomenological or something that you need to bring to the attention of the ASM. I think that major oil spills or those kinds of things have been used for these late-breaking sessions. But we wanted to use this forum, and we being the NIH Human Microbiome Project, to really announce and kind of highlight the work that's been going on with the Human Microbiome Project. It's a 5-year program. We're nearing the end of it. And just this past week, in fact, earlier this week, 2 major papers came out in Nature in the Thursday edition. And also there's a collection of 14 other papers that are called companion papers that were published in the PLOS journals. And they're called the Virtual HMP Collection, and they were linked to the 2 Nature papers, and they were all released at the same time. There was a lot of press about it. It really is the culmination of many, many years of work of the HMP Research Consortium. 200 members strong scientists across 80 different institutions came together to work on this project. So we wanted the ASM, which is obviously the best home for this major microbiological story, to kind of highlight what we did in the HMP. So on Tuesday morning, 8:15 to 10:45, ASM has offered to host a late-breaking session. It'll be the latest news coming out of the HMP. We'll also have a roundtable at the end, about 45 minutes. Jeff Miller, who's the president, ASM president-elect, has offered to moderate that roundtable. So we're going to talk about what's the future of the field. And then ASM also offered a separate Q&A session from 11:30 to 12:15. I think it's going to be in the press room. The idea there is to be able to entertain.
It's going to be here.
Oh,
ASM Live.
Okay, it's going to be here. That's what you're referring to.
Yes.
Okay. And it's going to be, questions will be fielded from the audience, from not only the online audience, but the live audience, as well as any press that calls in. So we'll have a 2.5-hour session from 8:15 to 10:45, and then a 45-minute Q&A session from 11:30 to 12:15 to really celebrate the HMP publications.
It's going to be really exciting. So all of our listeners out there, please join us again for that session. And in addition, I'd like to remind you that tomorrow at 11 a.m. Pacific Daylight Time, we're going to have another ASM Live, and we'll be talking about the role of non-food animals in the spread of antibiotic resistance.
Oh.
A very important and timely topic. So please join us again for that tomorrow. And I'd like to finish by thanking our guests, Lita Proctor from the National Human Genome Research Institute at the NIH, and Forrest Rohwer from San Diego State University. Thank you very much.
Thank you, Stan.
Thank you.
Microbes in the Natural History of Our Planet with Microbes in Evolution, the world that Darwin never saw. Available at estore.asm.org.